Thanks so much for introducing me. Thanks people out there for watching. It's about 5:45 p.m. in Chicago, Illinois right now where I am. And I work for the Morton Arboretum as I'm a pretty new postdoc there. I just started my position last year. This is going to be my first time talking about this particular project, and I'm excited about it. And as you're going to see, it's definitely a project that's still in development. I'm also working with Andrea Dawson at Mount Royal University in Alberta, Canada, as well as John Tipton at the University of Arkansas in the United States. And we're estimating North American tree migration patterns after the last glacial maximum, which was like 21,000 years ago, and using some Bayesian hierarchical modeling. So first I want to talk about what tree migration is exactly, because that might not be obvious to everybody. So yeah, we know that individual trees cannot move from one location to the next. However, tree populations can move across space in response to, for example, changes in climate. So if the climate starts to warm, as it did about 21,000 years ago, what's going to happen is at the southern edge of your range, you're going to have adults that aren't doing so well as the climate's getting warmer and warmer, and the adults are going to die off. And so that's, and whereas you're going to have successful seed dispersal, hopefully, to areas north of your current distribution. And as the climate continues to warm, those hopefully are successful. And so what you expect to see over time is this population or kind of species distribution that can shift in latitude, longitude, elevation. So here's just an example. I just used horizontal lines to show you the southern and northern edges of the distribution of this particular taxon. I think it happens to be maple, but I'm not sure what time period exactly. But the point is that you can look at two different time periods. If my slideshow will let me. There we go. And you can see over time that this distribution is shifting north. As you can see that the green part of the distribution in the left map is within the bounds of the blue and red line, and on the right, it's moving north of that blue line. Okay, so how do we know that this is happening in the past? Well, we know because of tree pollen. So individuals are gonna produce pollen, and using a microscope, we can differentiate between genera. In some cases, we can actually tell what species a particular pollen grain came from, but in general, it's just identifiable to the genus level. So the trees produce pollen, that pollen gets dispersed by the winds, and hopefully it'll blow, well, hopefully, depending on what your objective is, it blows into a lake or some body of water, where it can then deposit into the sediment. And as it deposits into the sediment, and the sedimentary layers stay pretty layered, so you can stick a pollen core down there. A modern person, researcher, can go stick a pollen core or a cylindrical core down into that sediment and pull it up. And then you have these layers, and layers toward the top are going to indicate the pollen was deposited not that long ago. Whereas the deeper you go in the soil core, the further back in time that pollen grain was deposited there. And there are all kinds of advanced models they're coming up with to relate radiocarbon dating to actual calendar years. And I don't have time to go through that right now, unfortunately. And so let's say you do this for one particular site, and then you have this information for a bunch of different sites across a big spatial area. So now you have spatial and temporal data. And what you can do with that is hopefully you can see what this GIF is doing. And so you have a map of, this is just an example of Acer, which is the genus of maple trees. And the number at the top indicates how many years ago we're talking about. So we're about to hit 21,000 again, which is the earliest time period that we're interested in right now. And right now it's getting closer and closer to modern time. And that blue shape, those blue polygons at the top are going to be the Laurentide ice sheet that covered a big chunk of North America 20,000 years ago. And you can see as it's retreating, the distribution of maple, which is in green, is kind of getting bigger, wider, moving up in elevation, latitude. And so these are the kinds of spatiotemporal shifts that we're interested in. And the reason we're interested in this is because if we can learn a lot about how North American trees responded to rapid climate warming in the past, then that can give us some better ideas as to how to conserve trees under modern climate warming. So clearly somebody had to interpolate this data because we have a raster here and we aren't going to have that many, we're not going to have a site necessarily for every raster pixel or cell in here, especially since we have to get these sediment cores from like lakes and peatlands and stuff like that. So they're not going to be found everywhere. They're going to be kind of sporadic across space and across time as well. But anyway, the way that people kind of interpolate this, that way we can get this nice continuous surface over space and time, is inverse distance weighting interpolation, or they'll model it as a simple Gaussian process, which is just kind of a joint distribution of all the taxa that you're interested in over some kind of continuous domain like space and time. But this estimation is very slow. And because it's slow, I think this has been, analysis of these shifts has kind of remained pretty statistically straightforward, kind of simple. And we also don't tend to estimate uncertainty in these estimates at all. And so my team and I are interested in trying to put together a pollen model that's going to be faster and that's going to account for uncertainty, and that's going to be more flexible. And so we're looking at this paper, Linderman et al., that came out a few years ago. And they describe a stick breaking formulation with this polygamma augmentation that allows us to take these multinomial models, which means you have a bunch of count data for a bunch of different taxa, for example. So we're counting pollen grains for 10 or 12 different taxa. We're calling that a multinomial process, and we're going to try to model it. And Linderman et al. Figured out a neat way to kind of do a math hack to avoid having to do these MC-MC sampling that is like takes so much time. And so you can actually rewrite this Gaussian process to allow for conditional sampling of the process parameters using Gibbs sampling rather than having to do MC-MC sampling. So that can hugely speed it up. So again, you rewrite the multinomial as a series of binomials, which is why they call it a stick breaking formulation. And then you introduce this polygamma distributed variable, which is also just part of this math hack to try to make it faster, which is why we're calling it, why it's called this polygamma augmented model. And we're going to be using a bunch of fossil pollen data. So this is publicly available. Anyone can download it from the Neotoma Paleoecology database. And this is a database of paleoecological information, so a lot of fossil information. And for us, we're interested in fossil pollen. And so we download a bunch of pollen grain counts. We have it for a bunch of different sites, different taxa, and different time periods. And depending on, and so I want to tell you a little bit about the kind of size of our data set. It depends on what time period you're talking about, because we tend to count a lot more pollen grains for more recent time periods. And then when you go back really far in time, you tend to count way fewer pollen grains. But for our more recent time periods, it's over 850 sites, maybe around 1,000 sites for more recent time periods across Eastern North America, and then maybe like 30 sites for 21,000 years ago. We're looking at 12 different taxa at the moment. And again, this is to the genus level. And then we're going to be breaking it up into 1,000-year time periods. Some people break it up into 500-year time periods when they work with this kind of data. We might eventually do that in the future. That way we can get more fine-scaled information. Okay, so I'm going to jump right into some of these results. And like I mentioned, this is a new project. This is kind of a lot of these graphs and figures are hot off the press. We're still developing this model. But what we're finding so far is that the model is working really well and very rapidly. And I'm going to show you some results of just a spatial model that I ran. And so this is the spatial model only for the most recent time period, so last 1,000 years or so. And we're using each of these graphs is a different tree taxon. So you see Acer, Ulmus, Betula, Cyperaceae up at the top. So different tree taxa. And on the x-axis of all these graphs, you have the observed data, or in other words, just our data that we have. And on the y-axis, we have the model estimated predictions. So if the model is totally perfect, you would see these dots lining up in a perfect one-to-one diagonal line. And you can see that they're lining up really, really well. This model is working really well. And what I want to show you next on the right side of the screen is just another way to kind of show you the same exact thing. So we have two columns of maps. The left column is our data. The right column are model predictions. And each dot is a site of where we found a core for pollen grains. And then I'm just showing you an example of 4 different taxa, how well the data are fitting, the model predictions are fitting the data for one particular time period, and it's working really well. But again, we have these different we have these different sites, but we eventually want to be able to look to see how the sites vary, or we want to be able to do out of sample predictions so that we know what to expect in sites in areas that have not been sampled with a core, right? So we want to be able to make out of sample predictions, which we can do with the model that we have so far. And kind of just from today, my collaborator, Andrea Dawson, was just able to finish the first run of our spatiotemporal model. But she was only able to run it on a geographic subset of our data because otherwise it would have taken too long and we wouldn't have been able to show you today. And so I just want to show you this geographic subset indicated by the yellow box over there. An example of some of these out of sample predictions that we were able to make over time. So this time the columns are 3 different taxa I'm showing you, and the rows are going to be time. And so as we move down, time is getting further and further back into the past. That might be the other way around. But anyway, over time, as you scan from top to bottom, you're going to see these really nice smooth changes in the pollen estimates over time. And it is what we would expect to see based on what we know about tree migration patterns. So that's really promising. And I know I'm running out of time, so just take home. So far, the spatial and even the spatiotemporal polygamma model is performing really well. It's going much faster than a multinomial Gaussian model would go. At the moment, we can jointly estimate pollen abundance for about a dozen taxa across Eastern North America. For over 800 sites, it was about a 9-hour run. But for about 30 sites, which is what we have for like 21,000 years ago, it only took a couple minutes to run. And I didn't show you uncertainty, but eventually we will be able to show maps of how uncertain we are in our estimates, which has never been done before for pollen. And John Tipton, my collaborator, his package that he's working on to be able to create these models and analyze these models is under development, as it's still under development right now. And that's it. Thank you. Thanks to my my team and for the Morton Arboretum.